Okay, let's dive right into this explainer. Have you ever wondered why certain isolated groups have incredibly unique genetic traits or maybe significantly higher rates of rare conditions? Well, it all comes down to a really powerful biological phenomenon. Today, we're exploring how a simple geographical split can absolutely alter the evolutionary destiny of a whole population. We're going to break down the mechanics, the math, and the ultimate health consequences of population isolation. To really wrap our heads around this, just picture a massive, diverse pool of colored marbles. That's our parent population. But then, watch how that massive pool gets squeezed down into a tiny, restricted bottleneck. Suddenly, only a few specific colors, just a handful of blue and purple marbles, actually make it through to start a brand new colony. And that right there perfectly captures the absolute core mechanism of the founder effect. You have a new population built entirely on the extremely limited genetic blueprint of just a few founders. So, to keep us on track, here's our roadmap. First, what is the founder effect? Then we'll go through the 4 specific stages. Stage I is separation. Stage 2 is sampling error. Stage 3 is endogamy. And stage 4 is consequences. And finally, we'll look at detection and a real-world impact case study. All right, jumping into section 1, what is the founder effect? Basically, the founder effect kicks in when a small subpopulation separates entirely from a larger ancestral pool. Because the starting group is just so small, they simply don't carry the full, rich genetic diversity of the main population. And guys, this isn't just some minor shift. It is a fundamental, hard reset on their entire evolutionary trajectory. You can really think of this as the life cycle of an isolated gene pool. It perfectly maps out into 4 sequential steps, starting with separation, moving into random sampling error, then endogamy, and finally, those crucial evolutionary consequences. But before we hit stage I, it's vital to know that these founder groups totally break the standard rules for genetic stability. In biology, these rules are known as the Hardy-Weinberg equilibrium. To keep genetics stable over time, you actually need 3 things: an infinite population, totally random mating, and absolutely zero migration. The founder effect shatters all 3 of those rules simultaneously. I mean, the population is tiny, mating is super restricted, and isolation prevents any new genetic material from entering the mix. Which brings us nicely to section 2, stage I, separation and isolation. So in stage 1, a handful of individuals migrate or just become trapped. This could be, a flock of birds blown to a remote island by a massive storm, or a group of humans establishing a closed, isolated community. Whatever the cause is, the ecological or geographical isolation means gene flow from the mainland stops completely. The door slams shut. There is an absolute cessation of new genetic material entering this colony. And because that door is permanently shut, we see an immediate loss of variation. The new colony inherits only a tiny, extremely limited fraction of the original genetic diversity from their parent population. To put it into perspective, if the parent population had 100 different versions of a specific gene, this new founder group might only carry 2 or 3. The genetic deck of cards has literally just been stacked. Let's move on to section 3, stage 2, random sampling error. Now, without the giant buffer of a massive population, random genetic drift completely takes the wheel. Because the founder size is so exceptionally small, allele frequencies, which is just a fancy way of saying how common a specific gene variant is, well, they start to change strictly by chance. So this isn't natural selection favoring the strongest trait anymore. This is sheer, unadulterated luck determining which genes get passed on to the next generation. This dynamic creates a really dramatic binary outcome: rare allele amplification versus allelic extinction. Since the group is so tiny, a rare allele, maybe a gene that causes a highly unique trait or even a rare disease, can suddenly surge in frequency. Why? Just because one of the original founders happened to carry it. On the flip side, many super common alleles from the parent population might face total extinction in this new colony, simply because none of the founders brought it with them. It's a massive reshuffling of biological probabilities. Okay, up next is section 4, stage 3, endogamy and inbreeding. So what is endogamy? It essentially means having severely limited dating options. It forces reproduction exclusively among close relatives within a very restricted group. Think about it. When the borders are completely closed and the population is small, eventually everyone becomes related. This heavily restricts the gene pool through forced consanguineous mating, whether the individuals actually realize it or not. And repeated mating among close relatives leads to a really fascinating genetic outcome called increased homozygosity. Scientists actually measure this with something called a rising inbreeding coefficient. I know that sounds super intimidating, but it simply means that offspring start inheriting large, completely identical tracts of DNA from common ancestors on both sides of their family tree. You are getting the exact same genetic instructions from both your mom and your dad because, well, they share a recent common ancestor. Which takes us to our final stage in Section 5. Stage 4, Evolutionary Consequences. So we have to ask, what are the actual stakes for the species when genetic variation drops this drastically? Like, what actually happens when a population is essentially playing with only a quarter of a genetic deck of cards for hundreds of years? Well, we end up seeing a devastating two-front consequence. On one side, you have inbreeding depression. This is the continuous exposure of harmful recessive alleles that directly reduce survival and fertility. Those harmful mutations, they suddenly have no healthy backup genes to hide behind. Then, on the other side, you have reduced adaptability. Widespread genetic uniformity totally destroys a population's capacity to withstand novel pathogens or environmental changes. Basically, if a new virus attacks and literally no one has the genetic variant needed to fight it off, the entire population is at massive risk. Finally, let's ground this in reality with Section 6, Detection and Real-World Impact. Now, you might be wondering how geneticists actually prove all of this stuff. Modern science relies on some incredibly powerful tools here. They use next-generation sequencing, or NGS, to literally hunt through the DNA and find those exact lost variants we discussed back in Stage 1. Furthermore, they use high-density SNP arrays to mathematically calculate the precise levels of homozygosity. This basically proves just how interrelated a population has actually become over time. And a perfect real-world case study of this entire four-stage process playing out in human history is the Amish population. The Amish experience actually perfectly mirrors that sequence we've just mapped out. They originated from historically strong founder events, meaning just a very small number of original immigrants. And for deep religious and cultural reasons, they've maintained strict isolation and endogamy over the years. As a direct result of that specific random genetic drift, they unfortunately exhibit much higher rates of specific rare genetic disorders compared to the broader general population. It's a truly profound living example of the founder effect's immense power. Looking at this entire journey, from that initial separation all the way down to the consequences, leaves us with a really critical question to ponder regarding the fragile balance of genetics. We know for sure that biological uniformity creates vulnerability. So without the deep genetic tools required to adapt, what does the evolutionary future hold for these deeply isolated groups in an ever-shifting world? It's an absolutely compelling puzzle of biology, chance, and survival.